opening up my thing. All right, thank you all so much for coming. Today I'll be talking to you guys about some huge overhauls I made to CZBiohub, which I'll go into detail quite extensively today, that would enable end-to-end interactive single-cell analysis. And we're calling this upgraded tool Excelbiohub, so Exploratory Cellxgene. So this workshop is assuming that people know generally how single-cell analysis pipelines work. But just to give a brief overview of what all these recipes typically look like is you start with two main modalities these days for single-cell RNA sequencing. It's either Cell Ranger for 10x genomics or SmartSeq2 outputs, and they're both in different data formats, but they will in the end turn into gene expression matrices, which are rows as cells and columns as genes, and then there are counts in there for how many of a particular gene you detect in that cell through sequencing. And then the next step is quality control, which involves all sorts of filtering. You filter the cells so that you remove empty droplets for 10x genomics, or you move contaminated libraries, or libraries that are just much lower in counts compared to other cells. You can also filter out genes. There are some genes that just may not be detected in your data set, meaning they have no counts in any cell. So you would just remove those. And genes that are like too highly expressed in every single cell of the data set, also tend to get removed because they don't really, these could be housekeeping constitutively expressed genes that don't really encode much information about the cell types. After you do some sort of quality control, you have a new matrix that you can then feed through a variety of different transformations. The most common set of transformations is to normalize each cell to have the same library size. So it's called sum normalization. And the target library size is typically the median library size of your data set. And here library size means the number of counts in your sample. Yeah, Sandy.
Sorry, Alex. That filtering of housekeeping genes, there are often splice variants of those housekeeping genes that tell you something funky. So you go all the way.
So this filtering, sorry for not clarifying, is solely for doing dimensionality reduction and clustering your cell types. When it comes to clustering your cell types, you really only care about genes that are expressed in subsets of your data set that help you delineate different cell populations from each other. If you were doing things like RNA velocity, you would be applying it to the raw data. RNA velocity would use the splice information that then it gives you trajectories. But in the end, you would overlay the RNA velocity vectors on top of your cell clusters, which are generated after throwing out these housekeeping genes. The other sort of transformation that people do to your raw counts is a log transformation, because the raw counts have an extremely heavy tail. It's following a power law distribution, which makes it kind of hard to analyze. You'll have some cells with some genes that have like 100 times more counts than another cell that is even the same cell type. So they would condense the distribution by log transforming it. There's other ways of doing that, but log transform has been the most popular, although people will say it's fraught with issues, but that's another story. The next step is beyond your filtering of constitutively expressed genes or genes that aren't expressed in enough cells. You also do an even further reduction of your dimensionality by doing feature selection. Feature selection is going to take around 2,000 to 3,000 total genes out of an initial 30,000 usually. And these genes are selected on the basis of their variability, typically. So highly variable genes are going to be genes that are more likely to be variable across cell types. That's the assumption. So in the end, you've taken your cell by 30,000 gene matrix and condensed it to cell by 3,000 gene matrix. And then you feed that into dimensionality reduction and clustering algorithms. Dimensionality reduction is typically done through a combination of principal component analysis. And then those resulting components are laid out in 2D using T-as-an-E or UMAP. UMAP more commonly these days. And you can also do clustering on the reduced dimensionality to actually group your cells into clusters based on which cells are more similar to each other in the reduced space. Given your 2D projection, which I'll show you in the next slide, you can then start annotating these data sets. And then once you have your annotations, you can then do marker gene identification. Marker gene identification amounts to just doing some statistics to figure out which genes are statistically enriched in which cell types. And then you can go through and actually use that to help improve your annotations, perhaps. So CZBiohub is a viewer of single cell data that was developed by a Chan Zuckerberg initiative. And where it fits into this pipeline is these last two steps. It assumes that you have already done all your analysis. You have your lower dimensional space. You have your computationally generated clusters. And then it's a tool that allows you to basically, highlight different cells and annotate them based on some genes that are expressing. And I'll show you a slide now. So just to quickly speed through and show you what the CZBiohub actually looks like, it'll show you this 2D UMAP, and then during the demonstration, I'll show how you can select cells and annotate clusters with whatever labels you want. And so what I wanted to highlight on this slide is that CZBiohub is a one-way viewer of pre-analyzed single cell data. So the way it works is that they have a Python backend that's running a server. And this backend is hosting your single cell data that's stored in a particular data format called an AND data. The AND data is an annotated data frame that has all these different attributes to it. ANDdata.x contains the gene expression data. ANDdata.obs contains cell metadata. ANDdata.var contains gene metadata. ANDdata.uns contains unstructured data that still might be relevant to your analysis. And then there's more attributes that are less relevant. But this basically is a data format developed by the TICE lab that totally encapsulates all the various aspects of your single cell analysis. And this is what CZBiohub is interacting with. So my goal was to upgrade CZBiohub to actually unlock all other steps of the pipeline after getting your raw data out of your sequencers. So I want to be able to do quality control and filtering, transformation of counts, feature selection, dimensionality reduction, and clustering, annotation, and marker gene identification through the front end, because that would greatly increase my ability to iterate, feedback and explore the data, while also allowing people who aren't computationally minded to be able to do these sorts of steps. So compared to CZBiohub, exploratory CellBiohub actually now is no longer a static viewer of the data. It now directly interfaces and writes to the AND data on the backend to modify it through preprocessing and things like that. And so you'll see exactly how we're interacting with the AND data in the demonstration today. So the outline for today is to process some data at first, because I gave you raw data with no information in it. It's just the counts. And I'll show you how you can do that super briefly through my front end. And then, I'll go through more of the features later on. And then once we have something to look at, a UMAP, I'll show you a demo of the features that came with CZBiohub. These are the features developed by CZI. And then I'll show you a demo of the exploratory CZBiohub quality of life improvements that I added to help with the interactivity. And this, at this stage, will still be only viewing from the AND data on the backend. We won't be making any interactive modifications to it. And then I'll show you how we can use CZBiohub to actually perform interactive analysis. And so at this point, I'm going to just switch over to a live demonstration. Oops, okay. So for those of you who want to follow along, you should be looking at a screen like this, unless you've already jumped the gun and started doing some pre-processing. So just to show you quickly what we're actually reading from. So when you launch CZBiohub, you're basically providing to it either a dataset name, which ends with a .h5ad ending or a CSV ending. H5ad is the native file type for AND data. CSVs are just, CSVs that are output from SmartTig2. Or you can pass it in a folder. And for example, this folder called CellRanger1. This is the output of CellRanger, and the output always looks like this. where we have a matrix.mtx file containing the non-zero locations of your sparse matrix with all the counts information. It's just a compact way of storing this. You have features vector just saying what are all the genes named, and then you have a barcodes vector saying what are all the cells named. And you can feed it in this folder. You can even feed into cell by gene a folder with all these different data types mixed together. It'll automatically read them all individually, then concatenate them into one. So for this tutorial, what I fed in was just 2 H5AD files. These are annotations that cell by gene then at runtime loads and concatenates. So we have our viewer here. You might notice this irritating dot in the middle. That's because we have to tell cell by gene when there's no lower dimensional projection available, just pretend it's all zeros. In later versions, I'll find a way of just like making it more intelligently, just showing a blank screen instead of this dot. But for now, this is good enough. And so this is kind of jumping the gun here, but we need to have something to look at to explore the native features of cell by gene. So what we want to do is we want to click this button here. It says click to perform preprocessing. We'll check the preprocess button, and I'll go into much more detail about what these various knobs do later on. We're going to go to transformation, click sum normalize. This is going to make each cell have median library size. We're going to click log transform. Sorry, it's a little blurry, Sandy. And then this is going to log transform with the data. Then we're going to hit preprocess. These spinners show up indicating that it's doing something on the Python backend, and preprocessing has completed. At this stage, we still have no dimensionality reduction done. So what we do is we click the button to the right of it, which has a tooltip that says click to recompute your UMAP embedding. And then we're just going to hit run. We're not going to worry about all the knobs yet. Oh, and at any time, if you have any questions, let me know. And I don't have access to the Zoom chat, so if someone's asking, just let me know. Brilliant. So now we have a low dimensional projection of our data, and we can look at it. And we have a bunch of dots here. Each dot is a cell, and the distance between dots corresponds to their similarity. If they're closer together, they're more similar. And if they're clustered relative to other cells in your data set, presumably they are corresponding to a cell type. So now that we have this view of our data, what can cell by gene do? So first of all, I want to preface this by saying Chan Zuckerberg Initiative did an incredible job making an interface that is lightning fast. It is the fastest interface I've ever seen. It can scale up to millions of cells with no problem. And I have tried making interactive explorers on my own using like different plotting tools. They were all slower. So this is why I think it's so exciting to build on top of this to enable more features that allow more interactivity. Someone is asking what the second step after hitting this, you hit some normalized log trend, then you just hit preprocess. Sometimes there might be a little bit of a delay for the front end to communicate to the back end because it's all getting piped in through like this local networking interface, which in the future I'm hoping to change to be a more direct communication. So if there is a delay and it's taking forever, maybe just wait it out. And if not, that's a problem, and I'll have to look into that. So yeah, going through cell by gene's features. And so what cell by gene came with is the ability to load in any annotations that were in that OBS slot of the end data I showed you. The OBS slot attribute is containing all the cell metadata. So the only metadata here is this annotation vector called orig ident. This is automatically loaded by my cell by gene at runtime to indicate that there were multiple batches that were loaded. And here, the name of the batch corresponds to its file name. So there was one file named PBMC3K and one named PBMC500. And so you can click this droplet here, and it colors the scatter plot based on these annotations. So now you can see which cells are coming from which batch. And later on, I'll show you how you can use the front end to actually correct this obvious batch effect. You can see that these are both PBMC data sets, but their cells are completely separate from each other. And so this is something that you would use the front end to correct for. But for now, we're just going to use this view to just explore some of the interactive features of cell by gene. So not only can you color by these annotations, you can even create new annotations. So we'll hit the create new category at the top left here, and we'll give it some name. And we can optionally duplicate labels and cell assignments from existing annotations, but for now we're just going to leave it default. So what this does is it creates a new category called test, and currently all the cells are unassigned. So now what you can do is you can, there's this tool here to the left of this plus sign in the menu bar. It should be automatically selected at first. It's a lasso selection tool. So what you can do is you can circle a cluster, and then you can hit this plus button. Hovering over it reveals a tooltip that says add a new label to this category. And then you can say, we'll call it population A, and we'll say add label and assign 356 selected cells. So it's going to add the label and then assign all the cells that are selected. Now let's say I want to get all the other cells to be population B. I could just like lasso everything with my mouse pad, but it's a little bit annoying. So what I can do instead is I can hit this checkbox, which unselects all the cells. And then I can hit this checkbox, which reselects only the cells that are unassigned. Then I can hit the plus button and then call it population B, and then hit add label. So now we have a new label that we can color by, and boom. And so this is how people typically annotate these tabular data sets that they've been using cell by gene for. And they'll also use a combination of gene expression patterns and other things, which we will show in this workshop. So I already showed that you can select by annotations by unchecking these boxes. And so the beauty of cell by gene, the way it was constructed, is it's using this construct in JavaScript called crossfiltering. So what crossfiltering is you can have multiple kinds of selections that all intersect with each other. So for example, to show you an example of this, let's say I unselect only population A, and then I unselect urig-ident. So you can see that because I unselected urig-ident, meaning all the cells in this with PBMC3K or PBMC500 were unselected, you could see that A was also unselected because the intersection of no cell selected through urig-ident and only A being selected is no cell selected. If I turn on PBMC500, which are these cells at the bottom here, and you can tell by hovering over this label, it highlights them in the scatter plot. You can see, oh darn, still I'm not selecting the top cells because the intersection is still 0. So if I select the top cell, all of a sudden these guys get selected. So this is just to illustrate what it means to cross filter. And this can help you kind of filter out cells in really complex combinatorial ways. You can filter based on the expression of certain genes and its location in a particular part of your scatter plot, or its assignment of being a particular cell type. So that's cross filtering. And to remove the selection state, you see how there's the dash here? You just have to click that, and then everything is now selected. So that's another way of selecting. I showed you lassoing. The other thing that's really cool is I'm going to speed ahead real quick and generate some continuous metadata. I'll show you how to do all this stuff in a second. So let's say I have some continuous metadata. There could be continuous metadata that came with your annotated data frame. Typically, this is % mitochondrial content or number of genes that are detected in each cell. And these would show up on the bottom left here below the categorical metadata. But because I gave you an empty data frame, an empty data set, we don't have those. So in lieu of that, I'm going to show you distributions of gene expression. What I did here is I circled this guy, I did some differential gene expression, which I'll show you how, and it gave me a gene set. Each of these genes are presumably enriched in this cluster based on some statistical test. I think we're using Wilcoxon rank sum test. But this is just to highlight what a brushable histogram is. So these histograms are all brushable. You can, any histogram you see in CellBioGene, whether it's continuous metadata that's on the left or these genes, you can click any part of the histogram and then drag. And then it will select any cells that have within this range of gene expression. Because here, this particular histogram is reflecting gene expression. So you can see here, now we only have these top two left clusters selected and some at the bottom. Just to make it even more obvious, I'm going to color by this continuous metadata, which in this case is gene expression. There's a color bar that pops up here. High expression values are red, lower expression values are blue. And then we can see we're going to select the low part of this gene expression distribution. Here, this is actually a collection of genes. All these genes are combined together to make an average distribution. But we can also select individual genes and color by individual genes. So here we can click the droplet next to FTH1 that pops up here. And then we can click these arrows to expand it. And then we can brush this histogram as well. So all this was developed by CZI, and it is fantastic. I think this is the greatest thing I've seen for single cell exploration, single cell viewing of your data. So how do we do differential gene expression? So what we do here, I'm going to delete these just so we can start fresh. Let's say I want to find genes that are enriched in this cluster on the top. So what you do first is you lasso this cluster. There's other ways of doing the selection as I showed you, but for now we're just going to lasso. Then we can hit 1 at the top here. So the tooltip for this is to save current selection for differential expression computation in set 1. And then if we want to compare to the rest of the cells to do marker gene identification, or you can compare it to just another cluster to do 1 versus 1 differential expression, I'm going to lasso everything else. And then I can hit cell set 2. And then you hit this button on the right. It says see top 10 differentially expressed genes. And then these gene sets pop up. 2 gene sets. The first one is highly expressed genes in this population. And the second one is highly expressed genes in the rest of the cells. So coloring by the top 2 high, we see that this guy is actually lowly expressed on average, which is good. It means our differential expression is working as intended. The first one, you can see it's brightest here. The other thing with these genes that show up on the right here, which I only just discovered recently, is that you can actually show scatter plots between 2 genes. So let's say, I don't know, we have a couple genes in our set. Let's see, this one seems to be enriched. That one seems to be, okay, so these should have the same scatter. So we'll hit the X. In this case, it's CD779B. There's a little X there that we hit. This is going to say we want this to be on the X-axis, and then we hit a Y on another gene. And then at the bottom, what shows up is a scatter plot of their expressions across the cells. This is kind of useful for seeing how correlated is a gene that you have newly discovered with like a marker gene that you know about. And so this is super useful. And we hit remove at the top right of that to close it. And the last thing I want to show is a button at the top right, at the top of the menu bar. When a category is colored by, show labels on the graph. So this is going to calculate the centroid of each category. Here it's a little bit less sensical because we're coloring by batch and not by like clusters. But you can see that it overlays a label. So this is the end of the features that come with CellBioGene that I wanted to highlight. And everything after this is things I've added to improve the quality of life, particularly for analyzing massive cell atlases, because that's what we at BioHub are super interested in. So to do this, this isn't a big atlas. So I have preloaded a massive atlas, 480,000 cells. This is the Tabula Sapiens that BioHub helped create. And in the Tabula Sapiens, we have a lot of different metadata. We have tissue types, which you can color by here. We have the sex of the donor. They're nicely mixed. Thank you, batch correction. We have the donor themselves. We have cell type compartments. And by the way, I forgot to mention these stacked bar plots here show you within TSP1, what is the composition of their compartments? Because we're currently coloring by compartments. We can also color by sex, for example, and you see that each donor is just one color because each donor is one sex. Same for cell types. For cell types, we have, I would say like around 100 cell types. And we can, this is where we start running into problems with CZBiohub because we can color by cell types. And then you can see acinar cell is purple and adventitial cell is almost the exact shade of purple. So, and like, hovering my mouse over this thing, I can't really tell which purple I'm looking at. So this is where the centroid label tool comes in handy. This one next to the stars at the top. You click it and then, okay, I can kind of start seeing things. There's neutrophil here, kidney epithelial cells here, but there's a lot of crowding on your scatter plot. And this is no good, especially when you have annotations that are not generated computationally, which would be much more in line with this scatter plot, but they're generated maybe through expert curation. Expert curation annotations may not even be, they may be scattered throughout the scatter plot. And so the centroid doesn't really mean anything. And so this is the first tool, leads me to the first tool I added to CZBiohub. And this tool is this airplane. And I made an airplane because I kind of the name of the tool is LiDAR. It's kind of like a radius-based tool. And so it's LiDAR, and in keeping with it being like a radius-based tool, when you click on this tool, you switch from lasso selection, and now around your mouse is this circle. Using your scroll wheel, you can make the circle bigger or smaller. And you can then point to a particular region of interest, make a circle whatever size you want, and then click. When you click on it, at the top left, you see this metadata shows up. You can see that there's 8,278 out of 8,331 total kidney epithelial cells, and then there's also a smattering of other cell types that just happen to get mixed in there. So this is mainly a kidney epithelial cluster. I can go here, point, click. Oh, this is an acinar cell salivary gland. There's also 600 duct epithelial cells in there. And this actually allows you to now be able to very easily distinguish what cells are actually in a particular cluster you're looking at. And so this is the LiDAR tool. You should be able to do the same thing for this demo data set, although here it's kind of nonsensical to use. But here we see there's PBMC3Ks there. Here there's PBMC 500 cells. Going back, well, actually now we'll switch back to this data set. The LiDAR tool also is useful for people who may have trouble like seeing what colors. For example, if you're coloring by gene expression, maybe they have a hard time distinguishing between different shades of this color bar. So you can use the LiDAR tool to actually display metadata about the gene expressions within their radius of interest. So here you click and it says, okay, so for CD74, within the targeted region, I have mean expression 2 with standard deviation 0.8. Eventually, this is going to turn into a histogram that shows up to be a little bit more informative than just some text. But for now, this is still like a nice proof of concept. You can click there. You can click here. It's low expression. So this is really helpful for people who have trouble seeing colors, or in cases where the gene expressions are extremely scattered. Because a lot of times you have really bursty, speckly patterns of gene expression that make it hard to see concentrations on the scatter plot itself. If you're not coloring by any metadata, this simply shows you the number of cells that you're hovering over. This is useful, especially for the tabula sapiens, because there are so many cells in your data set that you have no idea what the density even looks like. Here I'm clicking and I don't know, let's say we look at the bottom here. There's 8,000 cells there. So this is useful for if you're just curious, what's the concentration of cells in a particular cluster. Although if you have annotations, the numbers are also shown on the left here. So that's the LiDAR tool. The other thing I added was something that is very near and dear to my heart, which are hotkeys. So if you are hovering over the scatter plot, you can hit Shift ? Shift ? There, I should eventually put a tool at the top that actually, click to display hotkey menu. So you don't have to remember what the hotkey is to display the hotkey menu. But for now, it's Shift ? And then it shows you some hotkeys that I've implemented. Let me zoom in here so you can see. And there's a few main things you can do with the hotkeys. So the first hotkey I want to show you is something that can dramatically speed up the rate at which you do differential expression analysis. So let's say I have a big data set, right? This is a small one, but let's pretend it's a big one. And then I want to do some complex selection. And maybe this complex selection is hard to get using like the categoricals on the left, because that's just easy. You just uncheck and check. And then I'll hit, cell set 1, and then the other cells are kind of, let's say, in a configuration that makes it really hard to like, I don't know, oh shoot, I messed up, really hard to select all of it. So what you can do actually is you can use this hotkey Shift I. So you select one, and then you do Shift I. What it does is it automatically sets the one you've selected to cell set 1, and then it sets the inverse to cell set 2. This is really useful specifically for marker gene identification. Like for example, let's say I have this is where it really shines. It's like I want to do marker gene identification for this cluster, and maybe I don't have an annotation for it, and it's a lot of point and clicks to actually generate a categorical for it for the sole purpose of selecting just that cluster. So what you do is you select this thing and then just hit Shift I. Now you have 20 cells in cell set 1, and then the remaining cells in cell set 2. The other hotkey, Shift 1 and Shift 2 are just the individual steps that Shift I is doing. They're setting it to cell set 1 and 2, but that's all done by Shift I. But it's good to have composable hotkeys. And then you Shift D runs the differential expression. So going back here, I don't want to do it on the big data set because it'll take forever. But I'll go, the way it would work is like, okay, I'll go this cluster and I'll do Shift I, Shift D. And then two things pop up here. And it was super fast. And you can now very quickly generate marker genes for any cluster of interest. So that's a differential expression with hotkeys. The next thing that I also found kind of frustrating with CZBioHub's cross-filtering was if I want to, let's say, do a complex selection, and then, oh God, my finger hit the edge of my mouse pad and I couldn't select everything I wanted to, then when I try to add to the selection, what's going to happen is it's just going to unselect the first thing. And so how do you add to a selection? Well, I have a hotkey now. If you hold Z to use multiple selection lassos. So now I'm holding Z, and then I can now circle this thing, this cluster I failed to get, and it adds to the selection. So this is another quality of life improvement that is super useful for adjusting your selection. Maybe you missed one cell that you wanted to include just for the sake of completeness. Otherwise, you would have to redo the whole lasso process. And then the last thing that I made so far, and now that we have a framework for hotkeys, anybody can make a feature request and I can make a hotkey to do it like really, really, really easy. So, but these are the ones I have so far. So let's say I'm just gonna make a bunch of gene sets on the right. And with a big data set of hundreds of cell types, you're gonna have a lot of gene sets that come up, because you have 2 per marker gene identification, one high, one low. So the only way to delete them without hotkeys is to hover over these 3 dots next to the droplet and then click delete this gene set. Hover, delete, hover, delete. And for hundreds of gene sets, it's kind of annoying to do that. So the other hotkey is Shift Q. Shift Q, sorry, zooming in. Where is it? Delete the most recent gene set. So this basically just deletes the gene set on the top, and you can just quickly spam Shift Q, blah, 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 it's gone. So this is also quite nice to clean up your interface. And yeah, so these are all the quality of life improvements I made to the front end itself. And now, if there are no questions, I'm going to move on to how can you actually use CZBioHub to analyze your data in an interactive fashion. So if anyone has any questions, now's the time to ask. Questions about CZBioHub, questions about some tools I developed, feature requests, suggestions.
Any questions from comments? I can pass around the mics, or Zoom people, just unmute yourselves.
Oh, great. Oh,
Probably beneath the level of this tutorial, but could you just walk through a very simple example case for me where you want to look for the top 3 cell types that have the highest expression of, let's say, ACE2, the gene?
yeah.
How would you go about, because that's something that, I might conceivably want to do with this software.
Yeah, so let's try that. So we'll create a new gene set here.
Gotta go to Tabula Sapiens.
Oh, right. This is the PBMC. We'll create a new gene set. I think it's, test. We'll call it test. ACE2. And then we'll optionally add a list of comma separated genes. I think it's all caps for Tabula Sapiens, so I'm going to try this. They removed the autocomplete feature, which I'm planning on adding back in. Although they removed it for good reason. It's kind of complicated to parse if you could do autocomplete.
Cell types that have the lowest level of ACE2 expression as well, in addition to the 3 highest cell types?
Yeah, so let's see how we would do that. I
Okay.
haven't done something like that, but maybe we can
All right.
check it out. So we'll color by ACE2. And then we'll try to brush for high expression. So what we'll do is we'll brush these guys up. And so it would be nice to have like, so when you're coloring by the gene expressions, this is also something CZI implemented, is you have a histogram of expression within each, you see instead of the stacked bar plot, you have a histogram. But because this histogram is like, there's so many zeros, because ACE2 is kind of sparse in the data, this all just looks like a bar, so it's kind of useless. That would be one way you could like just scroll through and see. Alternatively, a nice feature that would be good to add, which I am going to write down, is like a sort button. You can sort this, the ordering of these labels based on their colored continuous metadata. I think that would give you exactly what you want. The top 3 would be the top 3 expressing on average, and then you would just scroll to the bottom for the bottom 3 expression, or you would just hit the arrow and reverse the direction. Fantastic suggestion.
One thing that you can try on that view is if you subset.
Yes, so you can try subsetting. This is a button I'm going to show you later on.
And now the histograms in front of the cell types start to make sense. So you can quickly scroll through it and see which cell types have the most expression.
Yeah, so what happened here is you made the cross filter selecting only cells with high expression. Thank you, Angela. And then there's this button here that you can click to subset to currently selected cells and associated metadata. So it just removes out all the zeros, and you can just have you're left with these cells. Then what you can do, you can also just color by the cell type annotations, then use your LiDAR tool to just investigate what's remaining. I do like the idea of the sortable list too, so we can try that as well.
Can you go back to the subset that showed the histogram?
Yes.
That showed the histogram? Yeah, so you're looking at, sorry. So this is like high to low expression across like a single, how come there's like, those histograms look weird?
Yeah, so on the right, you see the histogram for all the cells. And yeah, so and for all the cells, you see that there's a ton of cells with like a huge bar at 10. That's because the default analysis pipeline for Tabula Sapiens truncates expressions at 10 units. In this case, units is 10 log 1 plus TPM or whatever. And so you have like a huge inflation at 10. And because of that, it kind of skews these histograms, first of all. Second of all, you can use this overall histogram to see what are the ultimate bounds. It goes from around 4.5 to 10. And so here, this is also 4.5 to 10, I think. And so
of the 37 cells in that class?
Yeah, of the 37 cells that have the appropriate range of gene expression, there are a big number with 10 and a small number with 4.5 based on this histogram. Yeah. gets a little tricky to use histograms intuitively when you have such sparse data. But if you had a gene set, which tend to be more continuous distribution, since it's an average of many genes, then these histograms would look nicer. And yeah, I wrote down the sortable category. I love that idea. Okay, cool. So do you have any other like requests for how you would do certain things? If not, I'm going to go into into more detail on actually the interactive analysis. So we're going to open up this pre-processing menu here. And so we have a lot of buttons that we can click. The first, the most important button is like, do you want to pre-process your data? Like, I don't know why you would uncheck this. Well, actually, you would uncheck it for a specific reason, which I'll go into. But if you're just unchecking it, and then you just wouldn't use this menu. But let's say you had 3 batches of data, right? And each batch you want to analyze or pre-process in a different way. Maybe one of your batches was already log transformed and filtered. So this is where this batch pre-process checkbox comes in handy. So you would click this batch pre-process, then you would choose your batch key. This is just the name of the categorical variable that stores this information. So we have org ident. And then we say, okay, so for which label in my batch key do I actually want to do pre-processing for? So for PBMC3K, let's say PBMC3K is already pre-processed. It's not, but let's say it is. Well, it is now, but when we load the data, it's not. And then now we can just leave this unchecked. Make sure it's unchecked because we don't want to do anything to it. Then we go to the other one, which does need some transformations. We'll click the other one, and then we'll check pre-process. And now it unlocks these options. These are collapsible windows just to make everything a little bit compact. And so it's not filtered in any way. It is, but let's say it's not. So we'll open up cell filtering. And then there are 2 options here. Eventually, all these options will have tooltips because right now they're just using ScanPy's nomenclature for what the parameter names are actually called, but they'll have tooltips. Min counts is saying how many minimum UMI counts or read counts does your cell need to have before you filter it out. And here, people typically go with like 1,000 counts or 500 or something. More advanced versions of this could also include like, this is something Way actually implemented in SE Friendly, which I got a lot of inspiration from. You can do like visualize scatter plots of these sorts of statistics to actually draw, or histograms to like be able to, in a data-driven manner, choose the right cutoff. But for now, we're just working with like, we're working blindly now. So we'll just say 1,000 min counts, which is pretty decent. And then the minimum number of genes detected. Minimum number of genes detected is just the minimum number of non-zero genes in a particular row of your data, where each row is a cell. So I'm going to leave this off for now. Gene filtering allows you to not filter the genes. And there's multiple options here. Minimum counts is what's the minimum number of counts across cells in total. What do you want to have before you filter out the gene? So we want at least 5 counts across all cells, or this is a number that you can play with. Alternatively, if you don't want to deal with that, you can actually just say, what are the minimum fraction of cells that you want this gene to be expressed in for you to keep it. So if you make it 1, everything that's expressed in fewer than 1% of your cells is all those genes are going to be removed. This is something that you have to kind of intuit based on the composition of your data set. If it's a huge cell atlas of 480,000 cells and you have a cell type of 20 cells, that's way less than 1%. So any marker genes expressed in that would actually get removed if you use 1 here. So you got to be a little bit careful with how you choose the parameter. But 1 is actually the default in most pipelines. So that's kind of funny. Like it could be really destructive because you just remove all those marker genes. And this one is what's the maximum number of cells that should be expressed in or detected in before you until you start removing these genes. So typically this is like 99. If it's in more than 99% of the cells, it's probably a housekeeping gene. So just remove it. That's gene filtering. And then there's transformation. Transformation is assuming that your data is raw data. And If you check this, it'll normalize each cell to have the median library size prior to log transforming, if you have it checked. Typically, these two things would work together because if you're sum normalizing your data, it's assuming that it's raw counts because it doesn't make sense to sum normalize things that are not raw counts. And if it's raw counts, you typically want to log transform it. So, but I left it, that they're individual knobs just in case. The last thing is that let's say you want to pre-process your data. You've already done pre-processing, but you want to redo it. This is where this orig.expressions comes into play because this stores the raw data that you had when you loaded your data set. If you want to process further an already processed expressions, maybe log it again for whatever reason, or do some things to it, you could choose X. X is andData.x, and it contains the most recently processed version of your data. But most often, you would just always want to pre-process from the initial starting point because it doesn't make sense to double log your data or do some wacky stuff like that. And so this would, all these options, let's say I do this, and let's say I make it like 5 or something, just to show you. Now, if I go to the other batch label, they disappear because all those options were specific to just this particular data set. Unchecking batch preprocess just leaves everything back to as if you're pre-processing the whole data set. So that's pre-processing, but we've already pre-processed our data. So we're going to not click pre-process. And then now we're ready to run our data. And I told you guys that we're going to correct for this batch effect. This is actually one of the initial motivations for interactive analysis, because I think Merlin and the zebrafish people were really trying to do this like subclustering of their data, which I'll show you in a moment. And when they're doing the subclustering, it's actually removing the batch correction in the process. So they're getting batch effects in their view in their subclustering. So that actually led me down this rabbit hole of just unlocking all these different options that you would typically have in the Jupyter notebook, but now from the front end. So now we're going to open this menu. All the cells are selected. So it's saying that we're going to be doing re-embedding on 3,368 cells out of 3,368 cells. We're going to hit batch correct. And then there's multiple methods. Again, these all should have tooltips, but if you're familiar with Scanorama, you'll know they have like these different parameters, but I typically leave them to be default. And then we choose what is the batch key. It's again orig ident. And then let's say we want to name this new embedding batch corrected. So we type that in. And then there's a lot of different knobs here. Well, I'm going to ignore this left checkbox here because it's my personal single cell analysis algorithm that I developed in my PhD, which I wanted to include, but most people won't really need to use it. But if you wanted to, you'll see that it'll just deselect certain options that aren't relevant for that algorithm. But for now, we'll leave it unchecked. Shout out though, it's a really great method. So if you want to use it, go ahead. You'll find that it's clustering much better than your typical ScanPy workflows. But okay, so scale data. What is scale data? Prior to PCA, there's yet another transformation people typically do, which is to standardize your gene expressions to have 0 mean and 1 variance. This basically allows, makes it that PCA isn't biased towards genes that have high dynamic range of expression. Housekeeping genes can have anywhere from like 1 to like 100 expression, and this variability actually gets captured by PCA. If you standardize it and it has like high mean expression and also high variation, then it compresses it down to a smaller distribution that isn't so intoxicating for PCA. So that's what scale data is doing, but it works best on data sets with a lot of cell types. If you have data sets with, let's say, only 2 or 3 cell types, this can actually do more harm than good because it's actually lowering the signal strength of marker genes. Because if you have like a gene that's on in a third of your data set, then standardizing will actually make its signal weaker. There's like you can think about it and do some math to prove it, but that's my recommendation. Keep this checked only for big data sets with a lot of cell types. So you would scale your data. And then now this is the feature selection step I was telling you about, where you further limit yourself to the top 2,000 genes based on some metric. so all these tools are using this platform called ScanPy, which is a single cell analysis platform. It's like a hub of all these different analysis and pre-processing methods. And by default, they select genes based on their variability. And in the beginning, variability was very basic. It's just variance. Then people discovered that variance is not enough because genes with high mean expression tend to have high variability. So then they do variance normalized by mean expression. But then that kind of behaves weird with a lot of zeros. So then they do these like regression models to like approximate what is like what is variability when regressing out like a negative not binomial distribution, which models like all these zeros, whatever. In the end, like this kind of takes care of all that under the hood. All you have to give it is the number of genes that you want to select for and number of top genes. Again, this will have a tooltip. The number of bins is kind of you only play with it if you know what you're doing. It's basically each cell's mean expression, each gene's mean expression is going to be normalized against the expression of genes in its bin to account for the fact that different genes will have different dynamic ranges of expression, but we'll just keep it at 20 bins, meaning it's binning your mean expressions into 20 bins, but a little bit more in depth than I want to go. So we'll keep it at 2000. Now you go to PCA, and here are all the parameters associated with principal component analysis. Ncomps is just the ScanPy name for the number of principal components you want to use. 150 is a little bit much. Usually you want to go down to 50, but I'll just leave it at 150. Like these days, these methods are pretty robust to this parameter. This is something that I included for myself since depending on the PCA solver you're using, it can either make it faster, slower. There are some issues with convergence for certain data sets. If you have a massive data set that's sparse, you might want to use a different solver. But this is kind of for people who are more like computation biologists. Typically, you would never worry about this. Maybe I would actually even replace it with like a more intuitive, check this when you have this kind of data, uncheck it when you have that kind of data. That'll come along with all the tooltips I'm going to add. And here, a step I didn't really discuss in these analysis workflows, but it's crucial for single cell analysis and dimensionality reduction is constructing a nearest neighbor graph. This graph is an abstract construct that is like just a graph of nodes and edges. Each node is a cell, and there's a connection between two cells if they're similar to each other based on some distance metric. So these parameters here therefore mean how many neighbors do you want each cell to have? How many edges will be connected to each cell? What is your distance metric? Here it's default cosine, but you can use correlation or Euclidean. And then there are various methods for actually constructing this graph, all their own algorithms. So this is kind of choosing the length scale of your analysis. If you have a cluster of like 5 cells, and each cell in that cluster is going to be choosing 20 nearest neighbors, that cluster will be fused to something else. So if you have really rare cell types and you want as much granularity as possible, you might decrease it down to 5 with the understanding that other clusters might start getting fragmented. So this is kind of, but like these methods here, these methods here on the right all try to mitigate that effect based on some fancy algorithms. So, but in my 5 years of working with this kind of data, have never once touched this parameter. So I never really needed to touch it, but I'm including it for the sake of completeness. There are SAM parameters that we don't have unlocked because, we're not checking it. And there's finally a UMAP parameter. This is like the minimum distance between cells. You can make it like 0.1 in Euclidean space. That's the default for UMAP. ScanPy's default is 0.5, but we'll just leave this as is. Okay, so just to review and make sure that everything's selected properly. I don't think I changed anything. Okay, cool. So now we'll hit run. We have scale data on, we're doing batch correction. We chose the batch key. If you don't have any batch key selected, you won't actually be able to exit the screen. So we'll hit run. It's doing its thing. And with luck, you'll see that the batches are now properly corrected for. And voila. Now you have an embedding with cells, orange and purple cells overlapping each other because they ended up being the same cell type in the end. And now you just removed the technical artifacts that were keeping them separate. So that is your first approach analysis, okay? The thing that is really, really powerful about this viewer, and my favorite feature, is the ability to subcluster. So how do you subcluster? It's really straightforward. You just select your cells via the lasso tool or categorical selection or brushable histograms. Then you click this plus button, and now you see you're re-embedding on 1,300 cells. So we'll just leave all the parameters the same, and we'll click run. It automatically subsets to this data. And then what it's going to do is it's going to generate a new view of your data. This is now, so what is the difference between subsetting and subclustering? Subsetting is just removing all other points in the scatter plot that aren't part of that set. Subclustering is actually you're running through each step of this analysis workflow from the data. And the reason this is so powerful is because in doing this, you can actually, like PCA can now focus on variability between these cells, not cells in the full data set. And so you see that the name of this embedding is like some hashed name because we didn't choose a name, so it just automatically generated one. So clicking on this menu opens up an embedding choice menu. It says there are 3,000 cells in the entire data set. And now it's saying, okay, so I have this menu here, this embedding, we're already select on it, so it's selected. But I also have the parent embedding. This is where I subclustered from. So it's hierarchically organized. I can click this, and now it's showing me a different set of embeddings. It's going to show me all the embeddings that have the same parent, because also all the sister embeddings, because we're at the root node, all embeddings with 3,000 cells are going to be shown. So here is the unbatch corrected one. Here's the here's the batch corrected one. When we go to the unbatch corrected one, so let me zoom in again. I keep forgetting that it might be small on the thing. You see that the one that we subclustered disappeared. It's not the child of this embedding. It's the child of the batch corrected embedding. So this is a very good way of organizing in like a complex data set with 26 tissues like tabula sapiens, and you want to do all this subclustering and subclustering. This is a nice way of organizing, what part of the hierarchy are you in? Let's say I don't care about the batch corrected data and unbatch corrected data, so I'll just click this X, it'll delete it. If I go to the root, and root is just what is the original thing that you start with, and you always need root. That's why you can't delete it, because if you want to like start fresh again and you only have an embedding with like 1,000 cells, you can't like expand the selection to be more than the cells that are in the embedding. But anyway, yeah, you always have root, and then you can also delete batch corrected. So what this will do is this will delete both batch corrected and all of its children. It's a nice way of cleaning up, but I'm not going to delete it yet. You can also use these 3 dots here next to the X to rename our hashed thing into like, I don't know, pop1, and it just renames it. So this is really nice, because now what we can do is go here, and let's stop coloring by everything. And so I've already done some re-embeddings because it's going to take forever with this data set. But I'm going to show you exactly like what steps I did. This is going to be for all zebrafish cell atlas people that want to like go through their data with great degrees of granularity. So let's say I want to know how many cell types or subtypes, or are there any, within kidney epithelial cells. So I'm scrolling down, I've unselected all the cells, and I'm going to click kidney epithelial cells. I click these cells, and then I do my re-embedding, blah, blah, I click run, I choose some parameters, here I'm using Sam, because I'm biased. And then it'll generate, and I've named it kidney epithelial. So it'll generate this sub embedding called kidney epithelial. Yes?
There is a question in the chat that asks, would it be possible to indent those child embeddings?
Would it be possible to?
Indent.
Oh, that's a great suggestion. Yes, because yeah, right now it's kind of, they're all just in the same level. Yeah, I love that suggestion. Indent child embeddings. Thank you for that. So we're going to click on kidney epithelial. And here I'm going to just remove the selection state. So, okay, so if we go on the top and we look at this cluster here, and we like try to zoom in on it using this zoom tool. I think it's working like that. You see that it's kind of like a blob, right? you can't really tell, like, are there any clusters in there? And the labels we have just call it kidney epithelial. But now when we go into kidney epithelial subclustering view, oh my gosh, there's like 4 subtypes. And we can actually do marker gene identification here. So I'm going to show you how to do that. But first of all, I want to show you a couple other buttons I added that are really important for massive data sets.
So that blob you showed you now has these 3 little things on the side.
Yeah, that blob, we've reanalyzed it.
Right.
We've repositioned them in a low dimensional space. And because we're only analyzing kidney epithelial cells, PCA is having a much easier time of finding the genes that best separate out their subtypes. When you try to analyze the full data set at the same time, PCA is going to be dominated by global signatures of variation. These are variations that are genes that are different between tissues as opposed to subclusters or sub-cell types. This isn't even a tissue, this is kidney epithelial cells. And so the first thing we can do is we can start doing differential expression analysis to see, are these clusters legit? Are there actually genes in there? What are the genes? Can we start labeling them? But if you try doing differential expression, the backend has 480,000 cells. You know, differential expression won't use all those cells, it'll do the subsetting, but subsetting is a costly operation for such a large data set. So there's a button here that should be on the same row as this safe thing, but I messed up the CSS. But there's a button here with the 2 arrows spinning. For other people who have more zoomed out views, it might be on the top row at the left, but it just got shuttled down. So you'll click write current subset into the backend. So this is fancy words for basically trick cell by gene into thinking that you're running cell by gene from just 8,000 cells as opposed to 480,000 cells. So I'll click that. It'll reload the page. Now it's going back to the initial view. You can just click back into your embedding using this embedding choice menu. Okay, great. Now everything is like lag free. We only have 8,000 cells. We don't have to worry about doing analysis on this. You know, we don't have to worry about subsetting a 500,000 cell data set. You know, it's a lot more convenient. So now let's do use our hotkeys, Shift I to set to cell set 1 and 2, Shift D. It'll do differential expression. And then 2 things pop out. Let's expand this. First, let's color by the average, and we see, oh, cool. First of all, the top 10 genes seem to, on average, be enriched here. That already is a sign to me that this is probably a legit cell type or subtype or state of a cell type. And you can do the same thing for others. You know, let's actually go for this one on top here. Shift I, Shift D, quickly generate a gene set. We click that, and boom, you know, there's like a lot of genes that are enriched there. We can click the genes individually. We see MALAT1 is only expressed in that cluster. The things that are gray have 0 expression, which I forgot to mention. And so you can now start doing literature search. What is MALAT1? What is it doing? What is this cell type? It's a really rare cell type of kidney epithelial cells. And so you could create a new category, label it, and so on. So that's great. I could manually create categories, but what if I just wanted to cluster this using a computational clustering algorithm? This is one of my second favorite feature I added. It's the ability to interactively cluster your data. So there's a button here called leiden clustering. Leiden cluster is an algorithm that it's called the leiden algorithm, which basically takes this nearest neighbor graph that you've generated, you know, with a number of edges connecting each cell, and then it looks at concentrated regions in this graph and has a heuristic greedy optimization algorithm to group cells into clusters based on the structure in the graph. So we'll click it. And then now we have a nomenclature here. It's leiden underscore UMAP : kidney epithelial. So this is basically saying it's a leiden clustering. The parent embedding is UMAP. There's a child embedding called kidney epithelial, and it's with resolution 1. This parameter here is a resolution parameter. Let's the higher it is, the more clusters we'll get. So let's make it 3, just to show you what it looks like. And now we have resolution 1, resolution 3. So here we've colored, and it's kind of beautiful. You know, we have this thing in its own cluster. This thing kind of got into many clusters. If we think it's only one cell type, we can just, you know, we can basically just do this. We'll expand this. We'll click add a new label to this category, new unique label, blob. Add label, assign. And then we have this new thing blob, and these other guys have zeros now. We could just delete these labels because there are no cells, because we basically overwritten them. And so now this is something that I can now use this clustering to start annotating my data. Maybe this guy's actually should be reclustered because there's some, but you get the idea. Let's say I want to relabel blob to something else. Actually, let's say mallet1. It's 8. Edit this label's name. And then we can just say cool cell type, whatever. And cool. And so now the beautiful part about CZBiohub, this is functionality that was already there because they had an experimental version of re-embedding. You can, if you've generated something in a subview, you can go to the parent view and you can see exactly where these cell types are. So that cool cell type, it's up there in like some other part of the manifold. But darn, I overwrote the backend and I want to go back to 480,000 cells to look at the global view. That's where this button next to it comes in handy. Reset backend to the full data set. This is when you're done with that particular view and you now want to go back to the full thing. You don't care about lag anymore. So you click that, it resets the page. And now we still have all the stuff we generated from the subsetted data. And this is surprisingly really difficult to like make sure everything stays in sync. But yeah, so now we have, these cell types are located there. And you can go further. You don't just stop at one view. Let's say I have a bladder, I've selected bladder cells and I've done re-embedding on bladder cells. So now, let's say I want to cluster these bladder cells. I click leiden clustering, and then I can UMAP bladder resolution 1. I color by it, and I also have the original cell type annotations. These cell type annotations were generated by experts, or a combination of experts and de novo annotation methods like on class or other things. So we also have cell types. Here we can use a LiDAR tool. There's macrophages, fibroblasts, myofibroblasts, etc. Well, what if I want to, but then I see, okay, look, these macrophages here, they're in 3 subtypes. If I color by the leiden clusters, there's all these clusters that are subclustering the macrophages. So can I actually add a tool to CZBiohub to help facilitate re-annotation efforts? And the way I do this is you have these checkboxes here. You can select a category. I'm selecting the leiden clustering. I'm also selecting the cell types. And then it unlocks this button here. You can display a sankey plot from your selected categories. So now we have a sankey plot that's showing me the connectivity between leiden clusters and the cell types. And let's go to the macrophages. Indeed, the macrophage is subclustered into all these different leiden clusters. The thickness of the edge here corresponds to how many edges are connecting these clusters on average in the underlying graph. So this kit is really useful if you want to just check the sanctity of your annotations against the computationally generated manifold, where the manifold here is your low dimensional projection. Alternatively, if you just want to see like how good are the cell type labels by themselves, with respect to the manifold, you can actually just only check one category and then hit sankey plot. And then it'll actually check how well does it align to itself in your low dimensional space. Here you see the T cells and mature NK T cells are a little bit mixed, which makes sense because there's like an edge crossing them. So this is a sankey plot, and it's going to be really useful for actually trying to continuously refine your annotations. So I told you we can go deeper than just one subclustering. And there's a lot of cell types here, and maybe I might want to subcluster further. For example, there are macrophages here that seems to have a lot of interesting poles and structure in the low dimensional embedding. There's a little tail here. I know, Leo Proctor would crucify me for interpreting this UMAP like too literally, but we see that there's like some substructure there. So what I've done is I selected this, I did the re-embedding, and now I have a child cluster that I've called macrophage 1 for whatever reason. I click it, and now it shows me this. And let's say I do, first of all, I'm just going to delete these. There's a delete labels button. And then, so what I'm going to do is I'm going to do leiden clustering. And now I have clustering with even greater resolution. And then I can go up here and I can see back to the bladder view and I can see, okay, I've been able to cluster with a great degree of resolution. Now let's say I do this for every cluster in this view. I go to the fibroblasts and I do the same thing. And here I'll just show you these two examples. Now if we go open the fibroblast coloring, we see, okay, cool, we've been able to subcluster with quite a great view. But you'll notice that everything else is invisible because we only clustered in the fibroblast view. So this is where another handy feature I've added called fuse labels. You check the ones that you want to fuse. Here I want to fuse this guy with this guy, and eventually with all the other guys that I've subclustered, or manually annotated in a subview, something like that. You click fuse labels, and then you have a hashed, it was too complicated and the name would get too long to fuse these names. So I just hashed it out to be a unique name that you can then relabel. All cell types. And then you can color by it. And now you've combined them all. The names, in order to avoid conflicts, I add an _0, _1 based on like which label it came from. It starts from 0, goes up to the number of labels you're trying to fuse. But if you, in the end, you'd manually relabel all of these anyway. So yeah, that's fusing labels. And the last thing that I want to highlight is, what if I only care about this particular view of the data? And I'm like a biologist who wants to use Tabula sapiens to find structure in this particular subpopulation of macrophages and explore the genes and maybe port it into a Jupyter notebook and do some further analysis. That's where this text box comes into play at the top. You can enter an output file name to save your data, your current view of the data, to a file name. And so in order to avoid clutter in the resulting file, it'll actually delete all parent embeddings and only keep children embeddings automatically. So we'll just say this is macrophage 1. And you can put H5AD if you want, but it auto appends it if it's not present. H5AD is the file name. You click save button down here, although on you guys' menu, it would probably be at the top of the screen. And then data has been successfully saved. In the folder that you launched CZBiohub from, you have an ANData now that you can then load into a Jupyter notebook or reload into a CZBiohub session if that's the only set of data you care about. Just be careful that this data set you've saved has probably been pre-processed, so you wouldn't redo pre-processing. Otherwise, you'll double log your data and things like that. But yeah, that's all the features I've added. And oh, there's one last thing. So let's say I'm coloring by, whoops. Oh, there's a bug. Let's say I do differential expression. First, let me just write this set. Let's say I do differential expression. This is one thing I missed. It'll do its thing in a moment. There's this data layer thing. If you have other counts or other transformed counts that you want to visualize the expression of, or select using brushable histograms, you can actually choose which layer of data you want. There's an attribute in ANData called .layer. And let's say you've loaded in a data set that starts off with process data in X, but has a layer that has raw counts in there, like kind of like here, dcont X counts or raw or whatever. What you can do is, here, this is clearly log transformed. The range of expression is small. Then we can switch to this thing. And now the range of expression is huge. So you can actually change which particular expressions are you looking at. And this is similar to the data layer option in the pre-processing, like which data layer are you going to be processing from. Okay, now that is it. And so I think with these sets of tools and your suggestions, we now have a tool where Merlin or anybody who is intensively analyzing giant cell atlases can do every single step of the analysis pipeline using just the front end. And it's not just useful for people who don't know how to code. It's useful for anybody. Like in my PhD, I developed like a much more rudimentary version of this GUI, and it dramatically increased the rate at which I can explore my data sets. So I think this is really exciting. And thank you so much to the initial people who worked on cell by gene. CZI, like I said, they created an interface that is the fastest I've seen. Like, this is ridiculous. Like, we're loading a 500,000 cell data set, and then you can just, do filtering on this and display cluster annotations with like minimal lag. Although, as I say that, it kind of lags. But yeah, like, it's just beautiful. Like, look at this coloring. If I wanted to do this in Plotly or something, it would crash my computer. So thank you so much to Cell by Gene people for all their hard work and their beautifully constructed code base. I got very intimate with it in the last month or so, and I've had a lot of fun learning JavaScript for this project. So yeah, if anyone has any more suggestions, any questions, any like how-tos, please feel free to ask now. Or since a lot of you are from BioHub, you have my Slack. And if you're from CZI and you don't know my Slack, ask Colin, and he'll put you in touch with me.
Thank you so much, Alec. I just want to add on that there is also the link to the GitHub that Alec has been developing all the code. So for those of you that are on Zoom, and we have quite a lot of people on Zoom that are not from BioHub, feel free to raise issues there, or then you have at least my emails from the calendar invite. If you email me, I will forward it to Alec.
Does this then get incorporated into Cell by Gene, or how does the public get access to all of this?
So this is on a separate. Oh, sorry.
The quality of life and extra
things
you've added.
So all this stuff is there's too many changes to add them into the original Cell by Gene repository. Initially, I had just made hotkeys because Colin is the guy I've been working with mainly. He's my contact from CZI. Thank you so much, Colin, for all your help. Initially I was making hotkeys. Hotkeys are something that is really straightforward to integrate into Cell by Gene, and I opened a PR there, and that was fine. But like as I add more and more features, I've probably done things to the code base that they think is that real software engineers probably would think is blasphemy. So at this point, if we wanted to incorporate features into Cell by Gene's main repository, it would have to be on a case-by-case basis. And there's also, questions of like, if you put a feature in the Cell by Gene Git repository, someone from CZI has to also be involved in maintaining it. And so in the end, we decided it's best to just make a fork of it in CZBiohub's Git repository. That's the Git link that Angela mentioned. All changes are going to be going there, and maybe in the future, some of these features might make it into the on an individual case-by-case basis into Cell by Gene. But if you want to get access to this, it's already in a separate pip package. Those of you who followed along on the demo data set, you just installed it, pip install Cellxgene, and the GitHub is publicly available. So yeah.
Do we have more questions from comms? I can pass.
I did have a question, if you guys can hear me.
Yeah, hi.
Hi. Okay. Yeah, so I had a question, but so one of my favorite features about ScanPy, and this is over the Seurat pipeline, is that using their Git markers, their differential expression, if you can call it that, since it's just like Wilcoxon, but using that tool, they will basically do all the comparisons of one versus all for you without having to go loop through them yourselves and organize them yourselves, right? So then their visualization functions will actually just plot the heat maps and whatever you would like, and it would give you whatever number of genes you want from all the DE analyses instead of just one, right? Mm-hmm. So instead of having to rifle through tons of them, you can actually display large figures very quickly. Any thoughts to implementing something like that? Because doing one versus one all the time can get really time consuming, and plus organizing it in your right panel gets kind of long in the tooth as well.
Yeah, so we're already making progress towards the dream of like making these cell type marker gene identification easy, because we don't have to do one versus one. We just like do one versus all using the hotkey super quickly.
Right.
For sure, it'd be really useful to like have a button you click and it'll just generate differential expression gene sets. for the sake of, not cluttering this thing, only the ones that are highly expressed in the
Yeah.
cell type of interest. It would just do it for all of them. For example, if I hit cell type, you click a button, it does it for all of them. The one thing I might be a little worried about is just like from a user's perspective, it's going to take forever. So they're going to see a spinning wheel for like a long time. And like the point of interactivity is like quick 1-2 punch sort of thing, like do something, see something, go look further based on what you've seen, blah, blah. But it would be super straightforward to just add a button.
Right.
Especially for smaller data sets, it wouldn't take too long to calculate marker genes for every cell type. So that is written down and it's going to come in the next version.
Okay. Okay. Thank you.
Hey, Alec. So yeah, great work. This is a lot of really useful tools for analysis. And I was wondering, so for all the methods that you implemented, like clustering, batch correction, and so on, so the default parameters that you have here, are those the defaults suggested by the corresponding tutorials? Are those some parameters that you think are best for people? And is your recommendation that people just stick to those?
So yeah, most users would not need to modify these whatsoever. And so that's why I made it kind of, that's why I adopted this sort of collapsible thing. I don't want to show all these scary looking knobs if they're not interested in tuning them. And these are defaults that are chosen by ScanPy. So whatever wrapper that Scanorama people have developed through ScanPy's external tools package, I'm just using the same default parameters for those.
Oh, I see.
And I'm not showing all parameters. I cherry picked the ones that I thought would be most often tuned. So that's a little bit of subjectivity on my part. Most users, like I said, though, would not want to actually touch the pre-processing parameters. I'm sorry, the re-embedding parameters. Maybe for pre-processing, they would tune these a little bit. Although the one downside with the current implementation is that without a histogram, it's kind of really hard to select the proper threshold. There's other methods that automatically do it for you based on using statistics like empty drops for 10x data, which I might integrate into the front end. But ideally, we'd have for the pre-processing, we have histograms. For re-embedding, the only thing they might play around with is this min dist parameter and maybe the number of principal components. And scale data is also something they would use. So min dist would basically control the concentratedness of your UMAP. If you made it small, if you made it higher, it would be more spread out. Yeah.
And one more thing. So would it be easy to add, for example, something like diffusion maps or something if people wanted to do trajectory analysis using a different embedding?
Yeah, super easy. You would just add I would just change this to projection and have sub menus for whatever method you wanted.
Yeah, I think that'll be very useful.
Hey, I had a quick question. I don't know how much sense it makes. For the option that you have for neighbors, is it possible to kind of, it basically tells for a particular type that you have selected, these are the number of nearest neighbors, and it calculates the metric. Is it is there a way that kind of plops a small plot which actually shows a weighted graph like the sankey plot that you showed, where these are connected and the thickness of the edges are depending?
Yeah, I mean, that's a great suggestion. So first of all, to clarify, the nearest neighbor graph is between cells. So individual cells, if you tried plotting 480,000 cells network in a 2D layout, it would suck. But what you can do, and what people in the TICE lab have done and incorporate it into ScanPy, so it's really easy to pipe into this workflow, is things like what's it called? PAGA. It's abstraction graph analysis or something, where they basically coarse in this. They do this. They coarse in the graph. So meaning they sum up all the edges coming out of all the cells in the particular cluster. Then they weight the edges in some intuitive, nice way, maybe using a Gaussian distribution or something. And then they lay it out using a 2D layout algorithm. And then they actually take all the individual cells and have a heuristic for mapping it onto the coarse layout. they've shown that this is really useful for like highlighting like trajectories, because it's much easier to have connectivity between clusters that you know should be connected if you are just interpolating the cells between their centroids, as opposed to like hoping that UMAP doesn't fragment them. So incorporating PAGA would be super great, actually.
Yeah, even if it is just for a small subset of data instead of like the 500,000 cells, like for a subgroup, like if you can do that.
You
Because
probably
these things
still
are done for, at least from the tutorial, it was like you select a subgroup, you do the pre-processing analysis and kind of like show if it is even just for the subgroup of cells, you can
Yeah, on a single cell basis for like a subgroup, you probably wouldn't get much more information than just the UMAP. You would always like if we were to include a graph, you would just want it to be between like coarse nodes, because like there's a lot of noise at the single cell level. Even these network finding algorithms are random, like because they're approximations. If you tried finding 20 nearest neighbors in a brute force way for a 500,000 cell data set, you'd have to compute 500,000 distances for each cell. It's intractable. So these algorithms are stochastic. The networks will be cluttered and cloudy even for small sets of cells. I know because when I first started, would try to make those sorts of plots. So I think the best thing would be to coarsen the graph and show that instead. But I think it's a fantastic suggestion to show a network.
We have more questions in the room or on Zoom. I think that's it. Fantastic.
Thanks so much, everyone.
Heuristic greedy optimization algorithm. Awesome. I wrote that down.